So the promoter, right, is where we need RNA call to bind, and transcription factors, their whole reason for being is recruiting RNA call 2 and getting it set at that promoter. TF2s, right, they're the general transcription factors, known as general. They're generic. They're in every single cell. Every single cell that expresses genes has to have these same TF2 family members doing the same thing to promote transcription. All right. And so one of the reasons I like this slide is one thing that I feel like is confusing to students is how can enhancers be super far away? How are they regulating what's happening at, or influencing what's happening at the promoter if they're like a million base pairs away, right? So one thing I want you guys to do as budding biologists is to not actually think that this is what DNA looks like in a cell, right? A cell is not a two-dimensional thing with PowerPoint images in it, right? It's three-dimensional, it's moving, molecules are always in motion. And the way DNA is packaged, it's wound around these histones and then all funky, three-dimensional conformations. So something that's many millions of base pairs away might be wrapped around in such a way that it's distance-wise really in close proximity to the promoter. That's the only way it's going to have an effect on something that's happening at the promoter, is that spatially in the cell, it can be positioned nearby, so that its recruitment of a tissue-specific transcription factor can have an effect. I get super distracted when I'm talking and it's in the light and then like spit comes out of my mouth. I'm like, oh. The professor up here like, rah. Okay. Try to tame it down. Over enthusiasm. Okay. So, all right. So let's talk more about this. So transcription factors, right? Regulate promoter activation. TF2s are always there. So let's talk about tissue-specific transcription factors. They're regarded as the determinants. They drive transcriptional determination of cell type, right? So you see variation in what tissue-specific transcription factors are expressed in different cells, in different tissues, right? And that's important for creating a different profile of genes that are expressed in one cell to the next, right? It's tissue-specific transcription factors in muscle cells that drive this, abundance of actin and myosin that get expressed, and then all the other proteins that give rise to the sarcomere, right? Those are specifically expressed in muscle cells to do that. You have specific neurotransmitter receptors that are expressed in different neurons, right? So how do those differences arise? Part of it is the cells have a different collection of tissue-specific transcription factors promoting expression of those different genes. All right. And so the reason that tissue-specific transcription factors bind to is the enhancer. So, right, because every cell in our body has the same DNA, that means the promoters that are there, the enhancers that are there are going to be the same in every cell, because DNA is not changing. What's changing is what tissue-specific transcription factors are present in those cells, right? And so sometimes people start to think about like the chicken and the egg question. Well, how, in the beginning, how do you get different tissue-specific transcription factors expressed in different cells, right? And that's something that is answered over the course of a semester in developmental biology. But the super short answer is that it happens when the differences are established really early on in embryonic development. So imagine a scenario where you have an egg cell, and the egg cell is really good at localizing different proteins to different parts of the cell. So say you have, we're going to choose my favorite signaling molecule, it's called Wnt. Say you have Wnt just localized to this part of the egg, right? Well, when the cells divide to form a two-cell zygote, only this cell is going to be exposed to that Wnt molecule. It's going to be exposed to a higher concentration of it. So already you've established a difference. because that Wnt, right, is a paraffin signaling molecule that initiates the signal transduction pathway in just that cell. Now you can get transcription in different tissue-specific transcription factors. So basically, the difference gets initiated early in development and then gets perpetuated. I'm not testing you on that. It's just I feel like people always have that question, and it's to encourage you to go on to the fascinating class that is developmental biology. Okay, so in this class, what you have to appreciate is that these different cells do have cell type transcription factors, tissue-specific transcription factors. They bind to these enhancers, and they help stabilize the transcription initiation complex, right? They help stabilize the whole complex of RNA Pol II with its associated TF2s at the promoter. Help facilitate that. They can do it in a couple of different ways. And so one of the things it can do is recruit histone modifying enzymes, right? So if you have a tissue-specific transcription factor that recruits a histone acetyltransferase, right? It's going to recruit an enzyme that helps open up that region of the DNA so that RNA Pol II and TF2s can better associate with that part of the DNA. That make sense? So that's one mechanism. Another mechanism is it can just help, as shown here, kind of help that complex be stabilized, right? It provides like extra glue, if you will. Okay, but no matter the mechanism, it's always helping get RNA Pol II to that promoter. Okay, so I'm going to walk you through this slide pretty slow. And this is a slide that actually is from a developmental biology textbook, because I think it does a better job of describing it than the Albert's molecular biology myself. So what are we looking at up here? Right, our PowerPoint version of a gene. So green represents the different enhancer regions for this gene. Red is the promoter, orange are the exons, and gray represents the introns. All right. So, and this gene is called PAX6. PAX6 gene encodes a protein called PAX6, which itself is actually a transcription factor. All right. So enhancers, DNA sequences that bind tissue-specific transcription factors can reside, right? UTRs, introns, near or far. So that part we have down. So let's learn some other sweet things about enhancers. So the tissue-specific transcription factor association with the enhancer is said to control the time. By time, I mean developmental time, or what time in when is it being expressed, right? Is it just in early embryonic development? Is it through? Is it just in adult life? Is it at particular times of the day, right? So time, when. So it controls the when of expression. Place. What cell? What place in your body, right? Where is it being expressed? And then efficiency. Who wants to guess at what efficiency of transcription means? How well, and what does how well like translate to in the context of a gene being transcribed?
How good the nuclear mRNA would be so it doesn't break down outside?
No, it doesn't affect the stability of the nuclear RNA, but good thinking. You see a
Rate of transcription?
Rate, yes, kind of has to do with it. So efficiency has to do with like abundance, which can get it rate. So how quickly, how well is it stabilizing RNA Pol II? Because the better it stabilizes RNA Pol II and releases it, the more RNA transcripts are going to get transcribed by RNA Pol II, right? The more it's efficiently recruiting RNA Pol II and releasing it. Go on, transcribe. Recruit another one, go, transcribe. Recruit another one, transcribe. If it's like, RNA Pol II, I'm trying to hold on, I'm trying to get you situated, and it takes a really long time, that's not efficient, so you would get relatively less molecules of inRNA transcribed. So instead of, Gravir, you were talking about, instead of it leading to like the prolonged stability of mRNA, it's really how many molecules of mRNA are you getting transcribed from that transcript, or from that gene. Okay. So time, place, efficiency. All right, some other fun facts. Many enhancers bind to more than one transcription factor. So right here, it's amplifying the pancreas enhancer, right? And it turns out that they've, in the lab, figured out that the pancreas enhancer actually binds to two totally different TFs, right? Two different tissue-specific transcription factors. And so, right, there's one that's called PBX1 that binds here, and one called MICE that binds here. Why might it bind two? Does anyone have a guess there? Why? Does it bind two? Did I hear regulation? Increased regulation. The more complex you make something, the more opportunities to regulate it, right? So I can think of two reasons, right? Maybe in one case, if you just have PBX1 binding, you get low levels of PAX6 transcription. Maybe if you have both of them binding, you get high levels of the gene transcribed, right? More isn't always better. In some situations, maybe you just want a little PAX6 expressed, whereas in other situations, maybe you want more. So having that requirement of, or the involvement of two different transcription factors gives you that modulation. The other could be, in other scenarios, maybe you absolutely need both to get any transcription, right, to occur. So in that case, say you have PBX1 also controlling expression of actin or something, right? Well, maybe you want actin expressed, but not PAX6. Well, the cell could express PBX1 and it gets actin expressed, and then you can modulate expression of PAX6 by then only occurring when MICE1 is expressed. So there are two examples. The take home is really more transcription factors binding gives you more possibility for complex regulation. Right? But same thing in signal transduction pathways, right? Remember I showed you the generic one where it was like red circle ligand binds receptor, and then green square, green circle, green triangle, right? It's always way more complex than that. So a lot of times, you know, when people are just learning about signal transduction, like, why the heck do you need like 30 intracellular signaling molecules? It's to provide more complex mechanisms to regulate that and modulate their response, right? We are complex organisms. Not too simple. More again this class. All right, so another thing that you might have noticed, oh crap, this gene has more than one enhancer, right? So a lot of genes, they're not just expressed in one cell. Maybe they're specifically expressed in 3 or 4 different cell types. Not all 200, but yeah, more than one. So in that case, they typically will have different enhancer regions. So, where is PAX6 expressed based on the name of the enhancers? Someone raise their hand so it's not like it all falling out. It's on the board. The enhancer, yeah. Where is it expressed? So tell me the names of the tissue that PAX6 is expressed in. It's on the board. The enhancers, yes.
The pancreas.
Pancreas is one of the tissues it's expressed in. What's another one?
Lens and cornea.
Look at you go! Yeah, exactly. So the enhancers are named after the tissues in which not just PAX6 is expressed, but so is the pancreas enhancer, is it also in the lens and cornea? No. It is, because all our cells have the same DNA, right? The pancreas enhancer is in every cell. But what's different about in the pancreas? The TSTF that binds that site is present, right? The protein, the actual tissue-specific transcription factor that binds green is present in the pancreas. And it binds right here to promote transcription of PAX6 in the pancreas. So, neural tube enhancer, is it also in the pancreas?
Yes.
Yeah. It's in every single cell. What's specific to the neural tube is a tissue-specific transcription factor that binds to this part of the chromosome, right? It binds right here. So this region was called the neural tube enhancer because that's where the tissue-specific transcription factor in the neural tube binds to drive transcription of PAX6. Right? And so having different enhancers, and so think about it. What are the odds that PAX6 needs to be expressed in the exact same time in all of those cells? Good. Those tissue types are really different and have different functional needs. Right? So by giving them different light switches, it means right here I have two enhancers. Right? So here and here. Well, right now I don't want this one expressed. So I have this light switch to turn it off. If I only had one light switch, that kind of sucks. Everyone's going to sleep now, right? So having the possibility to have different light switches, which is really what enhancers are, that association with the transcription factor and the enhancer, it's a different light switch, allows you to turn the lights on in different cells at different times when it's needed, right? According to if that transcription factor is there. Okay, with me? Okay, good. Because you have tests on this stuff on Monday. It's important. If I'm losing you, stop me, I'll go over it again. Yeah.
So if TFTF binds to the enhancer in that specific cell type as opposed to the protein, and if the TFTF falls off, if the enhancer region, then the protein will have the HTF. Okay.
Yeah. So, yeah.
Is it possible that having multiple transcription factors, specific ones, bind to it? Could it give it more opportunity to be transcribed, or is it usually you need both of them at the time?
Oh, so like you can find one of these, yeah, you can find both scenarios. Yeah, where you might need both totally to drive transcription, or maybe you only need one, but if you have both, you get more efficient transcription. Okay, right? So there's a lot, it's just those are two different ways to modulate transcription. So it could be a requirement of both. You're talking about like the pancreas example with you. Could be a situation where you absolutely need both, or maybe having both gives you better efficiency of transcription, and one you still get transcription but lower level. Allows you to modulate it a little bit more. Yeah.
So it says like any enhancer binds to more than one transcription factor, but is it that one transcription factor binds to just that one enhancer?
No, there are a lot of transcription factors that bind multiple different enhancers too. Yeah. It's all sorts of complicated. Yeah, great question. Yes.
So this enhancer region, that's considered one enhancer, right? So the highlight.
Yeah.
So since it's a pancreas enhancer, does that mean the blue highlighted region and the green highlighted region both function in the pancreas?
That sequence is in every single cell. It's just the protein that binds to those sequences are only in the pancreas.
Oh, right.
Yeah.
And so, but if it were to be activated by a transcription factor, it would function in the pancreas, right?
Wait, sorry. Rephrase your question. It's my fault.
Oh, so since it's a pancreas enhancer,
Yes.
and it was in the pancreas, and you have the tissue-specific transcription factor attached to it, it would still only function in the pancreas.
Oh, okay. Let me see if I'm trying to do my best at sleepy active listening. And what I think you're saying is, what would happen, right, if you took the tissue, if you took PBX1 in mice, right, the two transcription factors that bind, you took them and you put them into like a heart cell, right? They're not usually in a heart cell. Would they still bind that enhancer and promote transcription of PAX6?
Something like that.
Heck yeah. Yeah. I said heck. I did not say heck. So yes, right? And so there's a question like that on the practice exam. I have a question in like one of the slides coming up to kind of get you guys to think about that, right? So the question is, let's call the tissue-specific transcription factor that binds the pancreas enhancer well, let's just call it what it is. So say really, we need PBX1 in mice to promote transcription from the pancreatic enhancer, right? So say we want to express PAX6 in heart muscle cells. It's not normally expressed there. There are two different things you can do. One is what I just said. You could force the expression of those tissue-specific transcription factors in the heart muscle tissue. So you can force expression of PBX1 and MICE1. So, right, if these are the light switch, right, the transcription factors are the hand turning it on, right? So in heart muscle cells, usually PAX6 is off. It doesn't have MICE1 or MICE or PBX1 there. If you force expression of the right tissue-specific transcription factors in that heart muscle cell, well, you're giving it the hand to flip the switch, right? The DNA is just like, oh, I'm just waiting for something to bind me. Give it something to bind it, and it'll promote transcription, right? That makes sense? One other thing you can do. So really, again, the important thing to remember is that the association of the DNA that makes up the enhancer region and the tissue-specific transcription factor, if they are there forming that union and stabilizing the transcription initiation complex, you're going to drive transcription, right? So what if you took this piece of DNA, which is in every cell, including the heart muscle cells, maybe the heart muscle cell has a transcription factor called, I don't know, myostatin 6, right? Myostatin 6 binds a certain region of the DNA. Maybe its sequence is TAAT. If you create a binding site for that transcription factor by mutating this DNA, sweet, you just created you created the right light switch for a transcription factor that's already there. It's one. The take home is it's one or the other. You have to either provide the right light switch or the hand to flip the light switch on. Okay. Heart cell. So PAX6 is not normally expressed, right? And you have this enhancer region. So the two possibilities are force expression of PBX1 and mice, right? Sweet. Now all of a sudden you have the right pancreatic transcription factors that bind and drive transcription. So that one's the most, I think, straightforward, right? And for those of you who don't pick up things until after class, I can hear what you're saying to me in my head right now. Not easy to get that. Okay, you can force expression of pancreatic TSCS, right? The other option is, well, what if you just, I'm going to do it in, okay, so heart cell. So we have, I'm just going to be super lame and generic, and I'm just going to call it a heart TSTF, the tissue-specific transcription factor that's already there, right? So in this case, what I'm suggesting you do, it's the same enhancer. So in my example, this is the pancreatic enhancer. Here's, well, just create induce mutation in DNA sequence to promote right? So it's either giving the hand to turn the light switch or to changing the light switch, so a hand that's already there, you turn it on. That makes sense? Yeah.
So if we did the induced mutation, would that, the hard TSTF would capacity enhancer and then PAX6 would be translated, right?
Transcribed.
Transcribed.
That's it.
And then
That was the
not
that.
the hard, the original gene that it would attach to the hard transcription factor, right?
Whatever gene heart TSTF binds to,
Yeah.
it can still bind that.
No, but I'm saying that wouldn't be transcribed, would it?
Only when it bound, so, let me see if I understand what you're saying. So say heart TSTF. There's a specific heart enhancer that is upstream of some heart gene. It's still bind that.
No, but what I'm saying
If binding right here is only going to influence PAX6.
okay, that's
Because it's positioned next to the PAX6. It's positioned in such a way that it's going to regulate the PAX6.
okay.
Does that make sense?
Yeah.
So these are kind of the funky tools that molecular biologists use to force expression of things or to study how things are expressed. By either forcing expression of something in a cell or inducing mutations. Make sense? No? Yeah.
Theoretically, the pancreas not have it was induced and then successfully increased the expression of heart cells, then the heart cell would have to be
Oh no, this is just one gene, right?
like in theory that.
In theory, if you-
that protein would do the same thing it's doing in somewhere else.
Yes, yes, that is correct. Yeah, this part though, we're not even talking about that kind of far downstream stuff. I'm just talking about how you regulate transcription of genes, right? How you get the gene transcribed. And really, this is just kind of a thought activity to help you understand that it is just this, because right, what's happening is the TSTF is recruiting a histone acetyltransferase to that region of the chromatin to open it up. It can do that wherever it's at in the DNA, right? As long as you bring a TSTF and an enhancer together, it will drive transcription of that gene. Yes?
So for cells that don't need it, is there like a repressor that's on them to stop the transcription if it's not needed?
It kind of depends. So there are a lot of repressors. So we talked about transcription factors as activators a lot in this class, but there are transcriptional repressors for sure. There are, right, and those would be things that do things like recruit a histone deacetylase or, drive DNA methylation, which shuts off transcription. So yeah, there are a lot of ways to repress genes. So a lot of times it's not just the absence of a tissue-specific transcription factor. It can also be, additional repressors that are like safeguards to make sure genes don't get expressed. Yeah. Okay. You guys can ask me questions on this at any time in the lecture. So again, the mechanisms of how the tissue-specific transcription factors work is they can recruit modifying enzymes like a histone acetyltransferase. If you have a tissue-specific transcription factor that, whether it's here, or here, and it's recruiting a histone acetyltransferase, it's going to promote nucleosomes in what? The uncondensed or condensed conformation? Uncondensed, right? Open relationship. Time for transcription factors to come on in. Need some new molecules. Alright. Always wanted to stabilize the transcription initiation complex. So those are kind of the two founding ways that these tissue-specific transcription factors help open up the chromatin and help recruit or stabilize TF2s and RNA-12. Okay. All right, so I actually already made you do this on the last slide. So PAX6, right? If we're not doing any manipulations in a normal organism, PAX6 is expressed either in the pancreas, lens, and cornea, neural tube, and retina. And so again, why is it expressed there? What is it that's different about those cells? Yeah, the CSTFs are expressed there. Those enhancers are in every single cell because our DNA is the same no matter what. So on the quiz, that's when I asked it. I think there was a question about like enhancers are the same, and I was like, yeah, they're the same in every cell. The same. It's just are the light switches are there. It's just is the hand able to turn the light switch? Is that there? And it's not expressed in the heart because what isn't in the heart? Right, there's no tissue-specific transcription factor in the heart that will bind to any of these enhancers. Yes.
So since two different types of transcription factors can attach to that one enhancer,
Yeah.
for whatever reason, there was a mutation or something on like the blue side,
Yeah.
could the right side still work?
We have to do the experiment. But right, that's what I was saying is that I'm saying I don't know the answer for sure. That was like a funny way of saying it. But well, not funny to you, funny to me. Internal laugh really quick. So, and that's when I was saying that maybe there are two possibilities here, and I can look it up for this specific enhancer how it's regulated. But there are two potential answers to your question, and two potential reasons why an enhancer might have multiple binding sites. In one scenario, yeah, you mutate this. You don't have MICE1, you get no expression. Or maybe it's just, you can get expression with just PBX1, but maybe it's lower, right? You get a decreased stabilization of the transcription initiation complex, so you get less mRNA transcription. And that's really kind of the take home. When you have multiple transcription factors binding the same enhancer, it can either be an all or nothing, or they modulate expression. Thank you. That's a great question, and a lot of these concepts are difficult to wrap your head around when you first are learning them, for sure. At the end of the day, right, it's set up like this because of how, remember I talked about like at the beginning of class, a good indicator of species complexity is complexity of genetic regulation. Now you're getting a sense of, oh crap, yeah, it's really complex. Right? It is not this complex in a single-celled organism. This doesn't have to be. There's just one cell. You don't need tissue specificity. When you get to an organism that has 200 different cell types and just 24,000 genes, how do you get the different complexity? It's by having all these complex regulations of gene expression. Okay. Let's move on. All right, so this slide, this is a mouse embryo, right? And it's showing PAX6 expression in these two different regions. So this would be in the lens and cornea, and this would be in the developing pancreas, right? So it's not actually PAX6. PAX6 does not turn blue. What they've done here, I want to talk to you about a method that's used in a lab setting to study enhancers and gene regulation. What they've done here is used a reporter gene. So what they did, I'm going to show you exactly what they did here. They're trying to understand how the gene is regulated, right? So they took this region that includes the pancreas enhancer. This is pancreas enhancer, and then lens and cornea enhancer. And what they did using some sweet tools of molecular biology is they fused that to the open reading frame of a gene called beta-galactosidase. Or the gene is called LAC-Z, but it encodes a protein when it's transcribed called beta-galactosidase. This is an enzyme, so in a lab setting, if you add this substrate called XCal, it causes blue precipitate to form. this is an example of a reporter gene. The word is on the next slide. It reports on where another gene is expressed. It's a reporter because we can see it, right? So a lot of times in molecular biology, we're tasked with how do we see these processes? How do we detect them? And so we have all these cool tools, right? I've shown you slides that, like the image of the intestine, right? That was colored like red and purple and green, I think. That was a process called immunohistochemistry, a little bit different. It allows us to visualize these cells and visualize proteins. So beta-galactosidase, when you have expression of LAC-Z, you create this enzyme, it turns blue, right? So what happens here, so scientists, right, are trying to figure out, like, oh crap, where are the regulatory elements of this gene? So they chopped off this segment, tacked it onto LAC-Z. Of course, you still have to have a promoter, right? You need the site for RNA phosphatidine. And they just checked out in this mouse embryo, they added XDAL to it, and where do we have blue cells? We went, oh, we have blue in the lens and cornea and in the pancreas. That means this segment of DNA that we tagged on to LAC-Z must include a specific pancreas enhancer and a specific lens and cornea enhancer. If they attached this whole segment, where else would you see blue? Neural tube. Yeah, boom. You would see in the developing central nervous system all this blue. Okay, so reporter genes are used to detect or measure gene expression driven by regulatory regions. That's how we study these suckers in the lab. So the two most common reporter genes, there are other ones, but these are kind of the ones you'll hear about most commonly. GFP, which stands for green fluorescent protein. Here you can see it in this tadpole in the eye, right? So it's glowing green. And then beta-galactosidase, which you saw on the last slide, and also in this mouse embryo, you can see blue, right? And so fluorescent green and dark blue are not colors that you normally see in these organisms, and so you can rest assured that when you see glowing green, it's because the reporter gene that you introduced is being expressed. Okay, so in this case, what they've done is created, they've tacked on the open reading frame of GFP to an enhancer for this gene that's called lens crystalline protein. And so when they tacked on the enhancer for lens crystalline, they saw a GFP expression in the lens, which is where that protein is normally expressed. So this is useful if scientists are trying to figure out, oh, where is the enhancer region? They can take big old regions and start like cutting it up in different patterns to try to locate where the actual enhancer is. Or you can do things like measure if certain things affect the expression of lens crystalline, right? Like maybe you want to see if shining UV light for long periods of time on a tadpole either increases or decreases expression of that gene. You'd either see a decrease or an increase in the amount of green glowing protein, right? And the amount of GFP that's expressed. So reporter genes are a common tool used in molecular biology, and a lot of different fields of biology actually, to measure gene expression. In this mouse embryo, they've attached the MYF5 enhancer sequence to LAC-Z, right? And this is a gene that's expressed in muscle tissue, and so you can see it in some developing musculature in the face. And a lot of muscle tissue actually derives from these chunks of mesodermal tissue that arise on either side of the kind of developing neural tube. So that's why you kind of see it. It almost looks like vertebrae. It's not. It's kind of right next to where the vertebrae develop. The muscle tissue develops and then kind of migrates out from there, so it's populating more in the body. Yeah.
So we're adding the GFP gene to where we think that
Every cell.
to every cell.
Every single cell. Every single cell. You're you're not just guessing what cells it's expressed in, you're putting in every single cell because that lens crystalline enhancer is in every single cell.
Yeah.
The tissue-specific transcription factor that binds it is not.
Okay.
But this is what tells you, right? Where is that tissue-specific transcription factor present? Where is it normally driving the transcription of this gene? Yeah? Okay. Text box, what does it say? So, endogenous transcription factor. Endogenous. Anyone heard that term before? Who wants to tell me what it means? No. Okay. No? No? Okay. It's juxtaposed with the term exogenous. So exogenous is something that would be introduced from the outside, right? Exo, outer, from the outside. So from like a lab setting. So this is an exogenous gene because I've introduced it in a lab. LAC-Z isn't normally expressed in these organisms. And I've introduced it. That's an exogenous gene. When you make an artificial gene like this and introduce it to an organism, it's called a transgene. Exogenous, comes from outside. But we're relying on endogenous transcription factors to bind to it. Endogenous, already there, right? And endo, inner, it's already present in those cells. That's what we're really monitoring with these reporter genes, is where do the endogenous transcription factors that regulate those enhancers, what cell types are they in, when are they driving expression? You can ask all sorts of cool questions about that interaction between the transcription factor and the enhancer. And the reporter gene, what it's really doing is telling you where that enhancer has that interaction. It's not telling you really anything useful about the function of lens crystalline, right? Because the open reading frame of lens crystalline isn't there. It's just telling you where the light switch is for it. And the hand that binds. Where's that hand and light switch interaction occurring? When, where, how much? Yes.
So, understanding being right next to each other would help it be expressed in the same cell, but I guess don't they each have their own promoter regions? So like how are they both expressed at the same?
Promoters are really generic. Promoters bind RNA pol 2 and they bind TF2. So you can have like a minimal promoter that TF2s and RNA pol 2 bind. That's not good enough to drive transcription though unless you have this tissue-specific transcription factor present. So sometimes if you have a minimal promoter, you'll get a little tiny bit of expression, but you really won't get robust expression until you have the tissue-specific transcription factor helping to stabilize RNA pol 2 at that promoter.
So these both be expressed that you need to insert like transcription factors for that gene.
You're relying on, so the whole point of this is, right, to ask, so I'm going to put this in this reporter gene. If you make what's called a transgenic animal, every single cell in this tadpole has this trans. And then what you're seeing is, okay, which cells glow green? And when you see green, that means that the tissue-specific transcription factor that binds that enhancer sequence must be present in those cells, because you're getting expression of the reporter gene. So if I was to change this to the PBX1 and mice sequence, where would we see green glowing? Pancreas, right? So it's just like the rest of your DNA, that transgene will be in every single cell, but it's only going to be expressed if you have the right tissue-specific transcription factor binding to the enhancer that you, right, artificially put in front of this reporter gene. So the way these are, the minimal promoters designed, basically, is to not recruit RNA pol 2 well enough unless you have the TSTF. Okay. That make sense? Yeah. Yes. Solid. Yeah. All right. See some of you understanding and some of you going, I will study it later and ask you a question later if I do not understand it later, and that's fine. So that was the kind of student that I was. I was like, I will study later. I always had to rewrite everything, And it was like in my rewriting process that I'm like, oh, okay. That it would either click or be like, teacher. So this chart, this is not a chart that you need to memorize this information. What this chart is trying to convey, right, is that there are all these different families of tissue-specific transcription factors, right? And so I talked about the Hox transcription factor family when I was talking about gene families. Remember how I had like the rainbow-colored Drosophila embryo and the rainbow-colored mouse embryo, and I talked about how Hox genes help regulate tissues in the anterior-posterior axis. You can do funky things like take the Hox family of transcription factors that drive expression of all the leg genes, put them in the head of a fruit fly, and you get legs growing out of their heads. Right? So that's one example of a tissue-specific transcription factor. These are all examples of really big tissue-specific transcription factors that have really specific functions in different cells throughout developmental time. And really the take-home here is that we have so many different cell types. So of course we have a whole crap load, to use a scientific term, a crap load of different tissue-specific transcription factors. Right? We need to have all this complex genetic regulation expressing different genes in different cells. Okay. All the light switches are there. They're all always there. That's the DNA. It's the different hands. You represent all the different hands. All right. Okay, my enthusiastic 3:51 p.m. students. So this slide is just a super stripped down look at this whole idea of efficiency of transcription, right? And it just the stability of the transcription initiation complex. How stable are all those proteins at the genetic regulatory region? How well are they recruiting RNA Pol II? How well are they positioning RNA Pol II just so as a promoter? It's really stable and they're doing a great job. You're going to have increased efficiency of transcription, which equals way more molecules of RNA produced. It's really meager stability, like it's just a weak association of those transcription factors. It's just going to be less efficient. You'll have less RNA produced. Sometimes you want that. Sometimes you just want a little RNA. Sometimes you want a lot. There's an ad campaign for something. It's not like a stick. I don't know. Okay. I can't think beyond what's on these slides right now. Okay. And yet I'm still trying. It's like a Kit Kat bar. Okay. So the stability of the transcription initiation complex is really determined by that association of those transcription factors. How well are they positioned? How well can they recruit RNA Pol II? At the end of the day, that's what efficient transcription means, more transcription. So in this case, right, which gene has a more stable transcription initiation complex? A. Yeah, A. All right. Switching gears. So now we're going to talk about DNA modification. Right? So in the very beginning of the lecture, we talked about histone modification. And in that case, we were talking about modifying lysine residues that were a part of the histone protein. Right? Histones are proteins. They're composed of amino acids. Lysine is one of them, and that's the one that happens to get acetylated or deacetylated, right? So that's histone modification. Now we're talking about DNA modification. And the DNA modification that happens is something called methylation, right? And it typically occurs on cytosine. So here's regular old cytosine molecule, and then when it gets methylated, you get the addition of a methyl group. That changes the structure, right? Change the structure, it's going to affect the way proteins might be able to associate. Okay. And so what we know so far about DNA methylation is it seems to always inhibit transcription. And it's really important as cells differentiate. So a lot of times the cells are differentiating, they're figuring out who they are, they're finding their place in the organism, and they figure out what genes they no longer need to express ever again. So a really efficient way for them to permanently shut down transcription, methylate the promoter region of the gene, and it is permanently shut off. So the cell doesn't have to think about anymore, like, do I want this gene, do I not? It's just shut off. So one famous example that's used during development, so there's a lot of changes going on in the early cells of the embryo, and it turns out we have a lot of different globin genes. At 6 weeks, the epsilon globin, that's epsilon, right? 3 letters, 2 fat people in the house, it's epsilon. Okay, we're going with epsilon. Epsilon globin gene is expressed. It has an unmethylated promoter. Gamma globin is not expressed, and its promoter is hypermethylated. Right, so there's a bunch of methyl groups there. That switches at 12 weeks. The status of our globin proteins change, the way oxygen exchange starts happening is different. And so what happens is at 12 weeks, this promoter gets methylated, this promoter is demethylated. So you shut off transcription of what I'm calling epsilon globin, and you activate transcription of gamma globin. Now, what's happening with methylation in the embryo is a little bit novel because you can see the reversal of methylation states. At some point that changes, and DNA methylation is regarded as a permanent change. You permanently shut off the gene, right? So in adult organisms, we don't really have active demethylases, which would be the enzyme that would demethylate, right? The one time you do see active demethylases would be in tumor cells. In tumor cells, you sometimes see these demethylases that were active in early embryogenesis, but you don't want them active now. You want these genes shut off. So say it's like a terminally differentiated cell. You don't want proliferating, so you methylate some of those cell cycle regulating genes. If you have a demethylase active, it's like, it demethylates the promoter. Now all of a sudden you're expressing proteins that are driving proliferation, right? They're taking the brakes off. You want them. So most of the time when you learn about DNA methylation, unless you're in a developmental biology class, you are going to hear that it is irreversible, because that is largely true. That's how cells normally use methylation, to permanently shut off transcription of a gene. Okay. So how does it inhibit transcription? 2 ways. So one way is, well, you're adding a molecule to these cytosines that's changing the structure. The way that proteins bind to DNA, they require specific structure. You change the way the nucleotide looks, you're changing the structure, right? Instead of it just being a hand saying, shake me, it has like this thing in the way. It doesn't know how to bind anymore. It's all protein DNA interactions, just like protein-protein interactions. They're very specific to the conformation of what they're binding to. So those methyl groups just get in the way. And the transcription factors don't recognize it anymore. So that's one of the most prominent ways that transcription is prevented. And there's also evidence that in some cases, DNA methylation can lead to the recruitment of histone deacetylases that help promote a kind of permanent repressed state of those nucleosomes. So it can promote heterochromatin. If you have a bunch of methylated cytosines and you recruit all these histone deacetylases, it can promote a heterochromatin state. Questions on that so far? All right. Here's where it gets really cool. So say a cell is dividing and it's determined like, I want to methylate this promoter. So to make it efficient, so that all of the cells, like if it's say an adult stem cell, right? But it's specifically like a neural stem cell. It's never going to need any of its daughter cells to like create mucus like a goblet cell would, right? So how does it make the efficient, make that efficient so that every time its daughter cell doesn't have to decide, what do I repress, what do I not? It can methylate the promoter and make sure all of its daughter cells inherit that methylation state. Here's where the CPG island comes in. Okay. This is awesome. It's 4 o'clock. You've been listening to me for an hour. Take a deep breath. Do what you need to do. Okay, stretch. You can't do jumping jacks. I invite you. Wake up. This can be confusing. Thanks for looking at me. Attention. Okay, so epigenetics, right, is, I talked about that way back on Monday. It's alterations to the DNA that don't affect the sequence. When you're talking about epigenetic memory, you're talking about that same type of change that's remembered by the daughter cells. It's inherited, it's heritable, right? How does that work? That's where CPG islands come in. We're coming full circle. There are 2 really important DNA, they're called methyltransferases, right? They transfer the methyl group to the DNA for creating the initial methylation status and then perpetuating it on the opposing strand, right? So de novo, what does de novo mean again? Anew, yeah, so de novo methyltransferase. And just to make it annoying, that's the bigger number, right? The DNMT3 is the de novo, it's the from scratch. The DNMT1 is the perpetuating, right? So it comes next, it perpetuates the methylation state. So de novo runs along the DNA, it's like, aha, I see a CPG island. And a CPG island is a bunch of C's and G's. The P just stands for the phosphodiester bond between them. So it's just a bunch of CGs. It's an island. CG, 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 CG. So that's what's recognized by this de novo methyltransferase. It's like, sweet, I'm going to do some methylating of these cytosines. So why do we care if there's a G there if the cytosine is being what's being methylated? It's very important for perpetuation of it on the opposing strand. For making sure the other strand gets methylated, because I will draw some more. Very exciting. If you have CG, 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 down here I have GC, GC, GC. So in here we have methyl group, methyl group, methyl group. Oh, when perpetuating enzyme comes along, goes, oh sweet, there's a cytosine right here. I'm gonna methylate it. Sweet, there's a cytosine right here. I'll methylate it. Here, I'll methylate it. That doesn't work out if instead we have, we'll make it to be funny, it's the caca in my household. The caca sequence. Now we have T's down here. Okay, cool. So maybe we get methylation up there, but when perpetuating methyltransferase comes along, it's like, oh, crap. Unintended. There's no cytosine down here. The only way you get a cytosine next to the one that's been methylated is if there are guanines up here, right? Does that make sense? Yes.
What happens to mutation in the CG island? So if one disease fits into like T or B, rather than T or B.
Yeah, it kind of depends. So the most direct and simple way for me to answer you right now is, without getting into the complexity of it, is, right, you could destroy the ability of that region to be methylated, which would mean you drive transcription. Or, like one of the genes that I study in my lab, FMR1, we want FMR1 expressed. People who have Fragile X syndrome, what happens is they have this mass of a CG repeat in the promoter region. When that CG repeat expands, it's methylated, gene doesn't get expressed at all. But the only way this gets perpetuated to daughter cells is if you have the G's here, because that's the only way you get cytosines on the opposing strand. Go back to the less fun. It's no longer cock-hops, just swoosh. Now we have C's here. Now perpetuating methyltransferase has something to methylate over here. It has cytosines. So only get methylate cytosines. The only way to get cytosines on the opposing strand is if you have guanines on the other strand. All right. So DNME and D. Why is that so hard? DNMT3, the de novo methyltransferase, methylates, performs the initial methylation, and then you get that pattern perpetuated. So if you have a scenario now, right, where the cell is dividing, the two strands separate, the DNA is copied, you still preserve those methylations on the cytosines, and then perpetuating methyltransferase can come along and make sure the copied DNA also has methyl groups on it. So both of those daughter cells are going to have, they're going to inherit that same methylation pattern. Yes?
So it says CB island, like tons of it. There's usually like a lot of methods. We get a presentation just one, this is just one, where it's slowly shut down.
So that's when Gruber asked the question of like, do I get into the complexity of it? So shrug your ears off if you've heard enough. But yeah, usually no. Usually it's a lot of methylation that is shutting off the promoter. So it's a CPG island. Like for example, in the gene that I'm talking about in my lab at FMR1, if you have like up to 50 of the expanse of the CG island, nothing happens. But if you get up to between 100 and 250, now you're shutting off expression of the gene. So yeah, it's typically a lot of methylation that's driving either.
The fragmentation effects depend how big the
Yeah, I mean, you know, I could start every answer I give with it depends in this class. Like there are a lot of things that are very context dependent, and absolutely this is one of those cases where it may be, you know, if it's at a very critical binding site of a transcription factor and you methylate it, right? Like you notice PBX1's binding site is just 4 nucleotides. So that's a case where maybe if you did have a change in the sequence, it would impair binding. So it kind of depends. Good? Ish? Okay. All right, so all mitotic descendants would maintain the same methylation pattern. And then one place where you see epigenetic memory happening in us is exon activation, right? So you've heard of this, you know females are XX. The X chromosome is a big old chromosome with tons of genes on it. The Y is pretty tiny, and most of the genes on it have to do with maleness. So women, we really actually don't want transcription of both copies of the gene of the X chromosome being promoted in our cells. So at some point in early development, every single one of our cells, one entire X chromosome is hypermethylated. The whole X chromosome is methylated from top to bottom, and it shuts off transcription of that entire X chromosome. Turns out there's a few examples of the very tip of the X chromosome, but largely the X chromosome, the way it is inactivated, the way it condenses to form what's called the bar body, right? This condensed dark spot that you'll find in every person who has 2 X chromosome cells is hypermethylation of the DNA. And so in this image, it's just showing an early blastocyst and then a later stage blastocyst, which is very beginning of embryonic development. And what this image is showing, what do you think the blue's coming from? Reporter gene. LAC-Z, yeah. It's LAC-Z. So they used a reporter gene embedded on the X chromosome to show activity. And so in this case, they embedded the LAC-Z into the paternal X. And so what it's hard to tell, but every single one of the cells of that early embryo is blue, and that's because you're getting you're getting activity from both X's, and then at some point in development, one of those X's is shut off. So in all of these pink cells, the paternal X has been shut off, and the blue cells, the paternal X is still active. And so this is a form of dosage compensation, right? It controls the dose of transcription that's occurring from the X chromosome, so that only one copy of those genes is expressed. And so the things that we know about X inactivation, very early on, both X's are active, then one becomes inactive via global promoter methylation. It's totally random. So the pattern of X inactivation, you cannot predict it from organism to organism, from embryo to embryo. It's random, or at least that's what it seems like right now. It seems totally random. And it seems like inactivation is irreversible. So what this would look like, right? In the beginning, both X's are active in the fertilized egg. Both X's active. We have so 2-cell zygote. We have a 4-cell zygote. In this case, the maternal's been globally methylated, so we only have paternal X active. Here we saw both. Here, maternal only. Now, because of perpetuating DNA methyltransferase, it's maintained. So you don't see a reversion, right? So as soon as the paternal is shut off and maternal is active, it's only maternal that's active, right? Paternal only, both. At some point, right, one of them is chosen, and then it gets perpetuated for the rest of development, right? So every female is actually, our cells are chimeric for maternal versus paternal X. Okay. The only exception that we've seen is in very early embryonic development. So when people started cloning animals, what they found is they take DNA from like a fully differentiated cell, right? Like Dolly was from mammary cell. And the mammary cell, of course, is going to have a specific methylation pattern, not just on X's, but all the genes, right, that aren't needed in the mammary cell are going to be shut off by promoter methylation. And so if promoter methylation wasn't reversed, when they did cloning, which is when they cloned Dolly sheep, they took that DNA from a mammary cell and put it into an enucleated egg and triggered it to divide. If methylation wasn't reversed, that egg would just develop into like a mammary cell, right? But that's not what happened. It gave rise to Dolly the sheep. So somehow there was a global, like a demethylation that happened in the early embryo to allow all that DNA the possibility to divide again, or to be transcribed again. And that's why when people are paying all these like hordes of money to have their animals cloned, they're pissed when the animal is born and it's different, right? Because for a lot of different reasons, but one of those reasons being, well, it has a totally different methylation pattern. Like calico cats, for example. So this is the clone mommy. This is Cece the kitten that was cloned from this mommy. The calico cats are all female because the pigmentation gene lies on the X chromosome. So they're all, if you've ever had a calico cat and it gave birth to kittens, they all look different because, right, her methylation pattern wasn't inherited. It started over in the embryo. So this clone kitten has a little bit different patterning than its mommy, which is why you can never, right, like actually bring back the exact copy of any animal. Okay. In your final 2 minutes, I just want you to discuss this with the person next to you. So this gets back to the whole idea of reporter gene and enhancer. This is important. I took the time to make an in-class activity about it. It's important. So this is a gene, I just picked a gene that's expressed in neurons. You want to figure out why it's not expressed in this individual with a neurological disorder. So the problem is neuralignment is not expressed. Your hypothesis is, I think there's something wrong with the enhancer. So you do an experiment, right? You create this transgene using GFP. And you take the enhancer from the person with the disease, right? not actually from like their cell, but you use fancy molecular biology techniques and make an exact copy of what we're calling the disease neuroligand enhancer and attach it to GFP. And then what you do is you compare that to a normal neuroligand enhancer. And you put that into a neuron in a petri dish. So I want you just to think about this for a second, right? In the minute we have left, because it's important for you to understand this before we take these. What would you expect to see if the enhancer was the problem? Would the neuron, would it glow green? You're putting this into a normal neuron, right? You put this GFP gene with the disease enhancer into a normal neuron. If it's a problem with the enhancer, would you see GFP? Right? Because the normal neuron, is it going to have the tissue-specific transcription factor?
Yes.
Yeah.
Sure.
So if it's a problem with the enhancer, that it's not going to be able to bind GFP, right? So if you do see green, when you put this disease neuroligand enhancer into this neuron, if it glows green, what would that indicate to you? The enhancer is not the problem. What could be the problem? Yes!
Makes sense.
Yes!
I see.
If any of you are confused by the words that just came out of these students' mouths, come ask me the question. Because I guarantee I like this question, and you might see how